4:30 p.m. Indian Standard Time. And today is the first day of this program. So I'd like to welcome you all. So I see there's a lot of participants who have joined in, and as we discussed in our free webinar also, that today will be an introductory session and it will be an interactive meeting where we kind of introduce ourselves, and also we will be introducing you to our platforms. So again, welcome to Bioinformatics for Next Generation Sequencing. Bioinformatics for Next Generation Sequencing is a collaborative course between MIT University Kolkata and Pine Biotech US. As you see, this course is an applied bioinformatics course focused on data science methods in application to the analysis of next generation sequencing data. The meetings or the sessions like today's session will be recorded. Okay, so the meetings or the sessions will be recorded throughout the program, and we will be emailing you the recorded sessions to all the registered participants. Not only that, along with the session video, we will also be emailing you the presentation, related publications, and material during the entire length of the program. So I suppose to all those people who are registered, participants of this program might have already received the presentation, today's presentation. Similarly, in the coming days for the next classes, we'll be sending you the presentation in advance, and also we'll be sending you, related publications that you can see and be more prepared, so that we'll have a good question Q&A session, and also the online material that we have that is there in the educational platform that today we will introduce you to. So all those things will be discussed in length in today's class. So again, thank you everyone for joining, and I'm hoping that more people will be joining in. And I would like to introduce my team here as well. Deepsha is there in the chat. Joe is there in the chat. So also we have Soham, we have Sarah, who are our campus ambassadors and who will be helping you in the chat. So please feel free to write in the chat and ask us questions, so that we will be happy to address all those questions that you have. So also, as I was saying, that many of you are joining in for the first time with us, and we are really pleased and we are really happy to have you here. So it would be really nice if we can maybe write one liner about ourselves, our name and what we are doing in the chat, and greet everyone, and let's start this community, start this classroom by knowing each other well. So during this whole time, I would really request you guys to write a few lines about yourself in the chat, and we'll be reading those and communicating with each other throughout this course. As I was saying, that today is our first session, and together, as we have discussed during our free webinar also, today's session will be an interactive session. So where we will get to introduce biology as data science. So if you see here, you can see focused, the image shows focus on 3 people. So let's see, how does a research lab work? That would be exciting to know, right? Because the data-driven research, or any research that has been happening around the globe, everywhere, has a system, the way it works, how research happens, what happens when you do a PhD, what happens when you join an institute to go do a training. Like here, Jitendra as saying that he is doing a PhD. So similarly, I wanted to everyone, we have here people who are undergraduates, we have people here who who are doing masters, we have people here who are doing PhD, and we have also faculties. So it's really great to have you all here. So we wanted to start off with, why bioinformatics training, why biology as data science, and why this is for biologists, to say the least. I have been asked this question quite often, that bioinformatics being a new field, and also like next generation sequencing is again a new field that has come up and is being widely used all around to do some quality research. Everyone today in today's world are generating a lot of data, and they want to analyze that data, and they also want to understand the meaning of it. So here is an example of how that process is, how that process happens. So the person who generates the data at the lab is mostly the student or the researcher or the postdoctoral fellow or the person who is actually doing the experiments, the wet lab experiments, and generating more and better quality data. That person is generating data using, all the techniques, all the cell biology, applying cell biology and molecular biology techniques, and generating the data from from preparing samples and then sending those samples, and maybe going to the NGS machine. And also they're trying to generate more and better quality data, so that the question that they wanted to answer can be answered with greater detail. Who's this person? This person is the person who looks into the data, right? I mean, I have generated a lot of data. I'm a biologist, and now what I would do with that data, requires, that's a big data that I've generated, and now this data requires some analysis. So we would require maybe statistics, maybe we would require computers to run those statistics, because these are complex data. So how would we do that? We go to a bioinformatician who has knowledge of computer science, who has knowledge of algorithms, who has knowledge of data. And who's the last person? Oh, okay. Now we have the data analysis done, and we understand what the data means. Okay, now we can take decisions, like what this could help us in, what kind of publication we can write about this data, what kind of intervention did we, what kind of discovery we made. So all those things come to the desk, and after that we you know try to understand the meaning of that data. We try to write about that data, we try to publish that data, we try to do multiple things, because that is a valuable data. Now we have done analysis on top of our data, and now that makes sense. So this is the way actually a research lab works. But what are the challenges? Well, the challenges are pretty critical, and those challenges are the reason that we are here. Let me explain you in more detail. So this person who is a biologist at the very beginning, as you saw, this lady who's generating a lot of data, is an expert about his own, about the experiments that she is doing. She understands the biology. She understands why she is doing this experiment and why she is generating this data. The person next to him, the data analysis person or the bioinformatician, who is an expert of algorithms, do know about his work, his algorithms, about the data, but does not know about the biology so much, does not know about the experiment so much. So when the person who is generating data goes to a bioinformatician or a computer scientist, there is a gap that you know the person who has been doing it on his own, who knows about his data, will be trying to tell that bioinformatician that, okay, this is the meaning of my data, this is the biology behind it, what do you think about it? The person who understands less of biology will be, it will be really difficult for him to understand every bit of it, the kind of knowledge that the biologists have, and to do analysis based on that. And this is so critical, because these analyses are important to understand the result. So it will definitely gonna affect the decision makers taking the right decision, because the analysis would be, would have less insights from a biologist perspective, or it's very difficult to to overcome this challenge. So there is this gap that we keep on talking about, that how a biologist can understand and do things by themselves, so that they can understand and make the most out of, most
Hi everyone. I think Mohit's connection is there is some kind of connectivity problem. Just give us a second. We are going to sort through this. Just stay here. We'll be sorting through this. Yeah, hi everybody. Mohit would be joining us very shortly. There was a connection issue and it is going to be resolved very soon. I have just had a word with him. So thank you so much for your patience.
Hello guys, am I audible now?
Yes, Mohit, you are.
Thank you. I'm so sorry about that. I think there's a internet glitch. So let me share my screen again. Is it? Can you see my screen, Deepsha?
Yes, we can see your screen.
Perfect.
Thank you so much.
Yeah, really sorry about that. I know. I mean, this was not expected, but somehow there was an internet issue. So as I was telling you about the research methodology and the research process and the gap, I mean, that is there in bioinformatics in general, and that has been seen in different areas of research as well. Last time, I mean, when we discussed, we saw the history of genomics data, how things started from DNA, and then knowing all about the nuclear DNA via the Human Genome Project that led to that data explosion that we discussed. So I mean, people who have joined in today, and this is something that is been going on. So we started way back, and once we discovered about the DNA, the Human Genome Project. In the Human Genome Project, we were trying to look into the entire DNA sequence of human, and it took us a while. It took us 13 years to understand that, and we were able to, you know, understand and the codes of human genome, and that changed everything. You know, at that point of time, after that, there was a huge data explosion. As many of the participants have just started their academic journey, I mean, it is important to understand the basics. So we were talking about the modern biology. In this course, we will learn both the basic concepts and the concepts of today's world. You know, as a student, we do want to learn and understand all of it, right? I mean, we saw this that after the human genome in 2003, it's been a while, right? It's been like 17 years after that. So it's a long time, and if you consider from 1950 to 2000, 20, it's really like 70 years. So 70 years of research, 70 years of all those great minds that worked towards this cause has led to this moment where we have, not only thousands, we have now millions. And why we keep on saying this, database and data size, without, like, many people don't realize it, or maybe many people do realize it, but they don't understand the, the depth of it, that how big is it. So we are talking about data in petabytes, okay, not in terabytes, not in, normal terms. It's like thousands and thousands of terabytes together. So we are generating data from individuals. Now we are not talking about, initially we started and talk about, single gene. We were trying to characterize those single gene during the mechanistic era, from 1950 to 1990, or maybe 2000 till up 2015. We were doing a lot of, these experiments, which were for meant for a single protein. And then we moved to a different, era, where we were able to generate lot of data with very less, price. So that changed a lot of things. But before we go into the course, I would really like to tell you guys one thing from my experience, is that I keep on asking about what would be a right formula. Modern biology, basic biology, things are changing rapidly, and so many types of data, and so many databases, so many tools. It just, sometimes is overwhelming. Sometimes many of us feel that, will I be able to understand this, because it's moving fast. So I keep on asking myself, what is the, right formula? What would be the right formula? Whether should I stick with the basics? I brush up the basics. I learn the modern biology as well. Many of you are, as I was telling you, many of you are undergraduates here. Many of you have just started your academic journeys. So the answer to this question is, I would say, it's a mix of both. How? I mean, we practice modern biology. Here in this course, we'll be practicing modern biology, but we keep on learning about the syllabus. We keep on learning about the basic biology all the time, going back to the books and learning about what is a DNA, what is a codon, or what is, ATGC, for that matter of fact, and what is, what all those nucleotides, what all those amino acid actually means, what what kind of charge they have, what kind of physiology they have. These all questions are important, I think. While going through this course, what we have seen is that many of us get inquisitive about those questions and go back and just Google. I mean, we just Google it and we learn about it. Okay, this is what it means. And many of the keywords that are important for us to understand, like the human genome, for example. If you write the Human Genome Project, there will be a Wikipedia page describing that. If you write about, Illumina, and there will be a history of Illumina, you'll see different number of articles describing the growth and how Illumina has evolved from different sequencing machine to the sequencing machine that have been used in today's world. So I think it's a journey that we'll be doing together, and we'll be really happy like to interact with you and to learn together, because in the end it's a collaboration that we are hoping for, a good collaboration where we learn together and we do wonderful things. So talking about doing different and wonderful things, we we started, the modern biology that I was talking about. The modern biology is very much high throughput. Why it is high throughput? Can you tell? Can someone write on the chat that why I'm saying that modern biology today is high throughput? I just gave you an example of how the history of modern biology, or history of genomic era has come from the starting of getting the DNA structure to generating individual samples from individual patients about their genomic information, and how their genomic information is influencing their their healthcare. Yeah, large data produce. Exactly. So it's real time and needs a large amount of data. That is absolutely correct. So we started working on, as I was saying, we started working on the single gene we wanted to characterize. Now we were able to see all the sequences, not just a single gene. Now we can see all the sequences in that organism. So what we have also understood, that these complex biological processes are interrelated. They are interrelated amongst themselves. There is so much, and there are so many connections within themselves. That was only We were only able to understand that by omics technologies. A lot of it came through by understanding different types of omics technology. Phenomics is a domain where we are understanding the phenotype. Genomics is the domain where we are looking at the gene, where we are looking at the sequences, we are looking at the individual nucleotides, we are looking at and we are comparing those data, we are looking for mutations, we are looking for variations. Epigenetics is such a trending, and what do you call it these days, is very hot term, because of the kind of exposure it is giving to the biologist about the understanding of how our genome, that 1% coding part, plays so many important roles in different kind of activities. DNA methylation is one of those activities which we are able to understand better through epigenetics. Transcriptomics, transcriptomics is something that we are trying to learn here together. So we are looking at expression table, we are looking at expression of thousands of gene together, not only one, not only two, not only 500, thousands of gene. We are understanding that how a person is reacting, how the person's proteome is expressed in different conditions. So as I was telling you, now we are able to see expression of all the proteins. Now we are able to see microorganisms that are present in our gut, right? Gut microbe. And we are also trying to understand the effect of microorganisms on different environment. Like, we are understanding the gut microbiota in much more detail. The omics technologies are used in healthcare, it's used in environment, in treatment, and it has application in so many fields. So I'm sure today, if we learn about this technology, it will definitely gonna make our tomorrow better. Why would I say so? Because we will be more aware, and we will be able to choose the right mix based on our knowledge. So to help people understand these omics technologies, and also to bridge the gap between biologists and data scientists, Pine Biotech started with a mission to simplify bioinformatics. As you see here, we have a small team. Bioinformatics is a US-based company that provides resources, solution, training for research and education in biomedical informatics. So since 2017, we have been developing innovative approaches to incorporate bioinformatics training, educational materials, biomedical specialization into undergraduate, graduate degrees around the world. Pine Biotech is a technical spin-off from University of Haifa, Israel, Tauber Bioinformatics Research Institute. In this picture, you see Leonard Brodsky. He is the director of Tauber Bioinformatics Research Center. And Ilya Brodsky here is the CEO of Pine Biotech. We have a small team, and we have also the founder, Dr. Tauber, Dr. Alfred Tauber here. So we have a small team, as I was telling you. Avi is our developer. Julia, he's in Israel. Julia is also a bioinformatician. She's been helping us in designing various courses. Me and Deepsha are in Kolkata, and we've been working on different courses. We've been working on other aspects. Joe is here today with us. He's a PhD in biochemistry, and he is also our curriculum developer. Joshua is a WebEx and UX developer. Sahil Sethi uh, is in the US, and he's helping us in bioinformatics projects. We have several collaborations, as I was telling you, with different universities in the US, in India, and also in different in all around the world. So University of Nebraska Medical Center, we are running a course with them. Tulane University, we have a collaboration. University of Haifa, from where we originated, LBRN Network. We have several collaborations in India also. We started with MET, we have CNCI, and also we have we have collaboration with FDA and a precision FDA, which is a challenge which we recently worked on, where we were trying to address the problems with bioinformatics data optimization and also on bioinformatics data reproducibility. Now, coming back to our program that we're going to start, that we have started today. Today, Bioinformatics for Next Generation Sequencing is a program that is meant for all of you here who wants to learn about bioinformatics analysis and the application of next generation sequencing. And as I already described, the importance of next generation sequencing and its data, and how data science and machine learning plays an important role in understanding the big data that comes from next generation sequencing and or high throughput. So in this program, I mean, we have wonderful tutors who will be carrying you throughout this, helping you learn this, and guiding you throughout this journey. I'll be your tutor. I'm an expert in machine learning, and I have a PhD in computational biology from Jawaharlal Nehru University. We also have couple of tutors from MIT University. Being a collaborative course, we get privileged to be with experts in different domains and fields. We have Dr. Chitrabrata Mal here with us, who has a PhD in bioinformatics. We have Dr. Prabhuta Gupta from your university, who has a degree in molecular biology and is an expert in genomics. And we have Dr. Joe Callahan here, who is our curriculum developer, and he has a PhD in biochemistry and keen interest in metabolism. So I would also like to give the stage to Dr. Joe here, who would like to whom I would invite to say a few words to the students here who might be interested in learning more about the domains that we discussed here, and maybe in metabolism as well.
Okay, great. Can you hear me? Can you hear me?
Yes, Joe.
That's true.
Okay. I'm gonna share my screen here just shortly.
Sure, sure.
Um, so let's see if this will work. Can you see a bunch of carrots?
Yes.
Okay, good. All right, well, hello everyone. My name is Joe Callahan and I'm the curriculum developer at Pine Biotech. My doctorate is in biochemistry. Prior to graduate school, I attended medical school and I'm very interested in the genetic and epigenetic regulation of metabolism. I'm just here to say hello today, but I wanted to let you inside the kitchen and show you a brief animation of what I'm cooking up for the transcriptomic series. This is going to be regarding the estrogen receptor. And anyways, let's get started with that. The actions of the estrogen molecule are mediated by the estrogen receptor, which is a dimeric nuclear protein that binds to DNA. It controls the gene expression of the receptor. Like other steroid hormones, estrogen enters passively into the cell where it binds to and activates the estrogen receptor. The estrogen and estrogen receptor complex binds to specific DNA sequences, which is called a hormone response element. This activates the transcription of target genes. Since the estrogen receptor enters all cells, or is on all cells, but that are expressed in those tissues, estrogen is only going to bind to those specific tissues. And so estrogen's presence is going to be dependent upon both the secretion and then also binding to a receptor on a specific type of tissue. Those tissues are going to be the ovary, uterus, and breast tissue. So as you go through the transcriptomics material, you will likely hear about the estrogen receptor. And so I kind of just wanted to present you with a little bit of information as kind of a introduction to those things. Mohit, do you have?
Yeah, thank you, Joe. That was wonderful, and I think this will be really helpful when we go through the courses.
Great.
Thank you.
Uh-huh.
So here, I mean, guys, if you have any question for Joe, you can always ask him, and you can reach out to us, and we can, help you answer those questions. May that be from biology, some basic concepts that you want to understand. May that be from data science aspect. May that be from machine learning and other things that you wanted to learn. So we'll be right here, and we are ready to help you. Just don't hesitate to ask your questions. So let me share my screen again. So yeah, as I was introducing you to the program speakers and the trainers, the next thing is that our class today. So as we promised, we would give you an entire course overview. We'll give you an entire overview of the platform and the analytical platform, and then we will have a Q&A session. So let's quickly go into that. So before we start, I mean, let's just understand what is an organization page is. So everyone who organization page is like a classroom. So all those people who have registered with us for this course will be having access to this page, which is you can see the link here. So later, I mean, we will have a demonstration of how this organization page is and what all facilities or what all features are there, like the forum, like the you can see your own progress, you can see the syllabus. Last time we did a small introduction about this organization page. Today we will again go and demonstrate what this organization page is all about. Thank you, Beepsha, for sharing that link. So first, we wanted to discuss about about a very important thing, which is our educational platform. We've been talking about these courses. We've been talking about the projects. We've been talking about large data. We've been talking about bioinformatics in general and all this omics technology. But where do we learn from? These are our books. This is the educational platform, and this is the URL to go to that educational platform. So we will be demonstrating you, and we'll be going into the platform, and we'll be learning about these courses and how we can maximize and understand from these tutorials, and do also hands-on exercises. As we saw last time, and also every participant here for this course will have a dashboard, which means that during the demonstration we will show you a dashboard that each of you will have with your login, where you will be able to get access to the courses, all your certificates, and also your progress that you will be able to see through the badges, and also you'll get to see the points that you score. And you could also upload these certificates in different platforms like LinkedIn and Facebook. So we'll be going through all that. So in the next, so in this class, we will also understand how the transcriptomic 1 course is designed, and how you can access this course and start working on transcriptomic right away. But before doing transcriptomics, we will show you what all courses that we have, and especially we always start with introduction to bioinformatics, and then transcriptomics 1 and transcriptomics 2, transcriptomics 3. So these are designed in a specific manner, in a specific way. So transcriptomics 1 talks a lot about the transcriptome, how many different technologies are there, what have been used so far to understand the RNA sequence and the expression, what all technologies like microarray technology is used to understand large data. We'll be learning about that. We will be learning about the technologies before that, and then we will jump into the transcriptomics data analysis and how that is being done using the tbioinfo server. So as Joe was saying, that every course, the transcriptomics courses will also include an introductory video, which you can see in the course itself. So we'll be having going to the demonstration, and we'll be having the demonstration with you so that you understand. But before we go and do the demonstration, I would like to tell you that there is a specific coursework associated with this program. As I was telling you, there are some important courses that help us understand and bring all of us on a same plate, on a same stage, where we start doing the analysis together. So introduction to bioinformatics is one of those courses which makes you comfortable with the platform, which tells you about the areas of research that we just discussed in much more depth, how bioinformatics is used in agriculture, how bioinformatics is used in healthcare, how bioinformatics is used in personalized medicine, how bioinformatics has a use in defense. So all the areas that bioinformatics is applied, and all there are several videos that gives you a lot of insight about what is actually happening and why we are talking about this data growth and why it is important to learn about this. Next, as I mentioned, transcriptomics 1, that talks about the basic transcriptomics analysis, how to build a pipeline. Then we move into differential gene expression, and then we do machine learning. So we'll be doing all these courses together. We'll be going through, we'll have these guided sessions all the time. We'll be there in the chat, we'll be there in the forum, we'll be there in your WhatsApp group or your LinkedIn group, so that you can have a smooth transition, so that you can learn with ease and also clarify all your doubts. As I was saying, that let's go to the website and check it out. I mean, let's have a demonstration of how things are and how to make the most and access the platform. So you can simply copy this link, and maybe I can put it in the chat as well. I think Beepsha has shared it. So we have several links here. Please go to these links and let's do it together. So this is the portal that I've been talking about. This is the book that I've been talking about. So how do I access it? So I just click here. Either if I'm a new user, I click register. If I'm already a user, then I click login. So here there are several options again. This is like a free login that anyone can make. So you can log in with all these credentials, or you can log in using your own account by registering with a Gmail or whichever account you use, whichever email that you use. So I would just log in like this. And if you have any problems in, getting access, login, please let us know. Beepsha is here. She's been, helping with that. So please let us know, and we will be able to, solve that issue for you. So here, I mean, let's see what is there in the introduction to bioinformatics course, because this is the first course. This is the introductory course that helps us understand the areas of bioinformatics and what is actually going on. If you see here, you will see an overview about the course. Introduction to big data bioinformatics. Bioinformatics is in healthcare, and translational bioinformatics. There is again a video. You can watch this video and understand about the content of this course. And there is also a curriculum which tells you about the different lectures that have been there. This all follows with a quiz. So this has 10 questions, and you will be given 10 minutes to answer those 10 questions. Once you do that, once you go through this whole course, and once you finish this lecture, you get a certificate. You earn a certificate that you finished introduction to bioinformatics course. So like this, when you keep on doing different courses, you earn different certificates. You can earn as many as 10 to 12, 12 to 14 certificates just by doing these bioinformatics or these omics courses, which are very interesting. So we do these courses. We also give our feedback and the review. You can see that we have got a very good rating overall through different participants, and many people have written about the course, how they have liked it. So you are welcome to see these. Please do the course, and then you can also would really appreciate if you can write a review also, which would help us to improve this course further. So this is the first course that I've been discussing about. So let's go to the intro. You can just click and go once you take the course. So it's also a free course which is available for everyone. So any of you or your friends who wants to learn about this and does not have time right now to go into a program, they can start taking this course and have develop an understanding that would help them to decide why bioinformatics, why they want to do bioinformatics, what is the current areas that bioinformatics has been applied in. So if you see here, I mean, there are several: defense, public health, agriculture, environmental sciences, treatment development, molecular diagnostics, personalized healthcare. And in many of these domains, like multiple industries are working, not only in the world, but a lot of them in India as well. So before academia was doing a lot of research in bioinformatics in India. But now with this data growth and like with personalized data that we are generating, not only from just the going to a clinic and doing those tests but through our watches, through our which tracks many of the health related features, like our heartbeat, like number of steps we walk, like all those Fitbits and Apple watches, and also those bands. So that's also our data, like, right? And that the data which is generated for you every minute, if you start accumulating that data, it would become a big data. So it's pretty critical and important, and if we can correlate with those data with our health and with our treatment, that would have a huge potential. So that's where the industry is also working. So I just wanted to tell you this because it makes sense, because it opens us up with a lot of opportunities as well, opportunities to get jobs, opportunities to do good and great research, opportunities to collaborate in these areas. So please try to do this course first. It's a free course and it's available for everyone whosoever has joined today's meeting and are a participants or not a participant yet. Introduction to Bioinformatics is a good course to start with. So let me just share you the link as well. Now, as you see, there are multiple courses, right? And I was telling you that you can earn a lot of certifications by doing these courses, not only just certification, but also the knowledge, the kind of knowledge that you would gain, because these are hands-on courses. And with the login, with the login for for the analytical platform, you will be able to finish these courses, do the practice, because they have all the component that is required, and it would be a great experience. So we'll be doing it together, so nothing to get worried about. But let's start with introduction to bioinformatics, and I hope we have our next class on Thursday. Many of you would be doing this course and have more questions about the course and about the different concepts that we have talked in that course. So after seeing this course, there's another important thing, or very interesting thing, called the bioinformatics project. So many of us are interested in bioinformatics project, because a project gives you greater insight, because you understand, you try to solve one problem, right? And while doing that, we learn a lot. That's the importance of projects. But that learning and that project could be mostly it could be very much essential, or it could have maximum impact on your understanding if you finish and understand the concepts behind it. The concepts come from the course, and the experience comes from the project. So how do we make these projects? It's something that I wanted to talk about in very brief. So as we discussed about these courses, now we're talking about the project. So what we do is that we take data and methods from published and digested. What does digested means? Is something that a good research has come up and people have validated it, and it has some real significance in the society. So those papers, those publications that have significant amount of data and that have made an impact, we chose those publications, and then we try to simplify it for everyone to understand that what does it mean, and how can we understand, and how can we simplify it for everyone to go through the same experience, same research experience. So what we do is that we create a project from the data, from the methods, and we replicate it using our own methodologies, which are available in TBioInfo platform. And then we build a project. Similarly, people like you who wants to do a project, who wants to do a new project, can also come up with their own idea. They can look into the data, then we can understand the methodologies that are available, and then we design a noble and independent project. Here, last time, as this is a slide which I showed earlier also, this program will include a number of research projects that you can join. But that research projects would make sense once we understand and learn the concepts and the courses, through the courses, that how do we do, data analysis with transcriptomics data, how do we do data analysis with different types of data, and what is machine learning, why machine learning is being used, and how machine learning is helping us in solving one of these problems. So let's look into a deeper, this was through our last program, where Dr. Prabuddha, Dr. Chittabrata, Dr. Harshata, and many of our students like uh, like Soham here, like Sarah here, have contributed to doing different kind of projects which were really exciting. I mean, it was amazing to work with different research projects, and we're able to do something which has never been done before. So we worked on global distribution of Mycobacterium lepromatosis as a causative agent for leprosy, which means that we looked into the genome variation of two different strains, one from Mexico, one from India, Bangladesh, or people where the strains have been very specialized. So the mutations are different for different strains. So we looked into that data. We looked into transcription factor binding sites and promoters which were influential in fruit ripening. So how do we manage to do these projects? We were able to do these projects, and many of the students who started with no knowledge were a part of this project. Now they have certification that they have done this project. So how does that happen? That happened through the same process. We learned together. We asked the right questions. We keep they asked us a lot of questions. We helped them in understanding the platform, and via doing the courses and doing the practice on the platform, they were able to do these wonderful projects which are still ongoing, and hopefully we'll be able to do some great research together and publish, or maybe do something really noble. So we have different projects which I described. One of the project is patient-derived xenograft models, where animal models have been widely used because they have been used for generating a lot of data. So from there, we could, we started the discovery of biomarkers for breast cancer. This was one of the projects that is available on the platform. So we'll be doing all those in much more detail throughout this program once we start working on the data. I wanted to pause for a while and tell you about the analytical server. So this is where we do we discussed about the books. Now we are discussing about the bench. So what is the bench? Bench is where you do the experiments, right? So this is the bench. This is the server. This is a server which is such a wonderful server because this is a high throughput server. So what kind of data you're analyzing? You're analyzing data in megabytes, in terabytes. So it's a powerful machine. It's a powerful portal. It's a powerful server. And all you can do from your web browser. From just a browser from our laptops, we are able to do these critical, important big data experiments. So it adds a lot of importance and lot of value because it's very difficult to collect these sort of algorithms together and then build a pipeline and then host it in a computer that has capability to run those pipelines and then do these experiments. Being a bioinformatician, I have faced this issue all my life, to having a good amazing computer, and also the kind of platform like Linux, or like different kind of Sun platform, Linux platform, Windows platform. and then optimize these softwares so that they run smoothly. It's a very heuristic, it's a very difficult task. We make everything easy. That is the purpose of this whole program. And also that's what I've said, that is also our agenda, to simplify bioinformatics. So we created this server, we created this platform to help people do their data analysis very easily, and not only focus on the data analysis and the methods, but focus on the research, focus on the outcome, focus on the things that would help us in, serving the society in a better way, so that we conclude those experiments with proper, information. We conclude those experiments that could be reproducible. So those are very important aspects in research or in biology. We'll also have a demonstration of this, but before we'll have a demonstration of this, I would like to tell you something about the file formats. So it's very important, about the file formats, because it's the kind of files that we'll be looking at. So there's nothing to be worried about. These are very simple. Like, many of you might have heard about FASTA files. FASTA files are nothing. FASTA is a simple way of representing nucleotides or amino acid sequence of nucleic acid or proteins. So this is a very basic format with minimum lines. First line refers as, comment line, which starts with a forward slash arrow, if you know, greater than arrow, and gives basic information about the sequence. Similarly, there's a FASTQ format, there's a GTF format. So FASTQ format was developed by Sanger Institute in order to group together sequences and its quality score. So we say it, PhRED quality score. So we'll be talking about all those in much more detail in the next class, so that you get comfortable. But just to tell you guys that you can Google these and learn more about these, like FASTQ, FASTA, GTF. GTF has the same format as GFF. So gene gene technology. Let me remember. Gene technology format, or gene transfer format. I think, yeah, gene transfer format is GTF. And also there are formats like SAM and BAM. The SAM format is sequence alignment map. SAM, sequence alignment map, is a text format. So it is used for storing same sort of, sequence data in a series of tab limited format with columns. So most often it is generated as like the human readable form of SAM is the BAM. Like, BAM is like a sister of SAM. So BAM is binary alignment map. So we'll also be talking about these in much more detail. So nothing to be worried about. It's just that I want you to get familiarized with these terms, so that in next time you would feel more comfortable. It's very easy. We'll look into each example in much more detail, how these formats look, what does they mean, and what significance they have on the analysis. And I would be happy to answer any questions, like if you guys have till now. So please ask on the chat. So the server is color coded. This is again to simplify. If you see colors while building a pipeline, which we will see in a moment, you would understand that which part it is talking about. So pre-processing will be in blue, mapping will be in red, quantification would be in green, and differential gene expression would be in yellow. So you see these boxes colored in different colors, you understand their significance. The T-Bioinfo platform that we've been talking about has been developed by keeping open source softwares and the softwares that were developed on the algorithms that were developed in Tauber Institute. So algorithms that are developed in Tauber Bioinformatics Research Center has a logo like this, if you see. So you will see different pipelines with different logos like Tauber. So that means that these algorithms have been designed in Tauber Research Institute. So this is how it looks. As I was telling you exactly, that it's color coded. So here you see pre-processing. Here you see, mapping, this is different color, and again different color, expression different color, and we will have a differential expression. If you have chosen something here, it would have been yellow. So you would be able to understand which step you are in, and what all these steps are. We will be talking in the next class, where we'll be doing a live, demonstration, where we both will be doing it together. So we'll talk more about the demo pipeline today. I'll show you a demo pipeline. But what is a demo pipeline? And we'll go into different sections also to show you what all are available. Last time in the class, there was a question about machine learning, that people who have understood the data types, like, we will do RNA-seq analysis, we will build a pipeline, then what? Then we will do machine learning. And how we will do machine learning? By using these pipelines. So here we have supervised and unsupervised methods, which we will be using throughout this course. As I was giving you example of a pipeline and a demo pipeline. For a demo pipeline, it's really simple. We start with demo pipeline so that all of you get really comfortable with seeing a pipeline, building a pipeline, understanding the output of the pipeline. For demo pipelines, you don't have to worry about the input files, but on the later stage, we will be giving you the input files so that you can build your own pipeline and run from the, building an advanced pipeline, something like that. So we'll start from very basic, as I was keep on saying. We'll start with demo pipelines, we'll understand them well, and then we'll move to the advanced stages. So this is the link. I think, let me share you. I think many of you will be getting the login credential for this. Whosoever have are the paid registrants will get the credentials, and then you will be able to use the platform, login in there, and so let us go to this server. This is the server, and here is the same thing. With your credentials, you'll be able to log in with your email address and your password. You press login and you're in. Now here you will see several sections. We are making some new sections like cancer biology and biology of aging, but these are well defined and well known sequence. The best part is when you hover your mouse over it tells you about the section and what it does: NGS data, mass spec data, structural biology data, and then data integration and modeling. So here we will start with transcriptomics. If you just scroll below, you'll see 3 demo pipelines that we will be doing together. And if I click on this directly, I'll see a whole complex pipeline that I can build from myself. But we will be first doing, as I said, we will be first doing the demo pipeline. Here in the next class, it'll be really interesting because we will be building a pipeline. As you see here, there is no input files, there is no output files. You can just start by clicking here, start. The input files are already uploaded. So one of the important thing to tell you is that this platform is AI guided, means that you will be given the path to follow. So if you see here, I see that Trimomatic is being activated now. I click here. Okay. Now I see PCR clean is activated. Okay. Now I see HiSat is activated. Really good. And also I will see also the input files that are there after the next step, also the output files that are there in the next step. So you can always click download and see what does those means. And also these sections, like when you click on these sections, they talk about the process that is involved in this section. As I was saying, that this is meant for not only for experts, but for everyone, for biologists, for students who does not know anything, can learn from these by learning by going through these small sections and learning about these. If you have any questions, we are always there to help you out. So this is how you build a pipeline, which we will be demonstrating in the next class in much more detail. But this is an overview to give you that this is a very simple, server, very simple to use. Anyone with some knowledge and some guidance we will be helping you out throughout this course, and we will be doing all these analysis together. So So about the activity certification and the progress, how we see about the dashboard, I would like to give the stage to Beepsha quickly. So she will show you in a couple of minutes that how you can access and have a proper, proper dashboard where we will be able to see your progress, where you will get certificate with your proper name and also other features that are associated with it. So I'll give the stage to Beepsha. I'll stop the sharing.
Thank you, Mohit, for the wonderful overview. I'll be sharing my screen now. I hope everybody can see my screen. Okay, so this is how a profile should look like. So I'm going to quickly talk about your profile and how you can update it and what you can do with it. So I'm going to use Sarah's profile as an example because she has a complete profile. So you can see that she has a profile picture, her name is complete, she has her social media links. You can see which level she is on, the Amity logo, and how many points she has gathered. Plus, you can see what activity she has been doing. So the reason we always ask everybody to update your profiles is because in the certificates which you get after completing the courses, as Mohit was showing, is the name, this is the name that will be there on the certificates as they are auto-generated once you finish every course. So let's go first to the profile and we'll come to the certificates. So this is the profile section. As you can see that, Sarah has given her full name and she's written a little bit about herself. So this is a very good way of portraying who you are as a person, your interest, as well as, letting other people come to know you. And also it's a very good way for others to come and collaborate with you. If you have your LinkedIn here, people can find you for your work and ask you to join projects and, for any other such opportunities. So here you have your activity on your profile. In your activity section, you can see everything that you do in our platform. And then you have the profile, which you can update one time. The next thing you have are the courses. So we see that, Sarah has completed so many courses here on the platform. So she has signed up for the machine learning for biomedical data on January 11th. So you can even see, which date you have enrolled it in, how many days it took for you to do a course. So this is something you all can do at your own pace and time. And you do not have to see what anyone else is doing. You can take your time and complete a course. So, and it will all appear here under your profile. And also you have quizzes and certificates. So we'll go to the quizzes first. So each of the courses, as Mohit showed again, has a lot of quizzes. So you can see how much progress you have made, how much time it took for you to complete a quiz. And you can scroll through all of these tabs, and the more quizzes you have done, it will show up here. So you can also see, how many you have finished, passed, and failed, and you can retake them. And the next thing you have are the certificates that I was talking about. So you see that Sarah has now 8 certificates as she had completed Introduction to Bioinformatics, and her name is right here. So if you do not have your names here properly, you can just update them via your profile section, and you can download these certificates by yourself. So you can use this and attach it to your CVs. You can put it on LinkedIn. You can share it in your social media channels. All of those things are there. And this is not the certificate that we'll be giving to you after the course completion, but as you go on with each of the courses and you finish them. And also there is one thing that I wanted to remind everybody here: once you do the courses, we get a lot of complaints sometimes that we are not able to generate certificates. So to generate a certificate, you have to finish a course completely and do the quizzes, and then click on the finish course button. There would be a yellow button to finish the course. Just click on it and the certificate would be generated for you. Sometimes it might happen there is a glitch, and we would of course be helping you with that. But in most of the cases, you just have to click on the finish button. So the next thing that I wanted to show you is the projects, which you will come here. So you can see that, Sarah has done the Spodoptera project. And as Mohit has showed you, we'll be talking a lot more about the project. And the other thing that I wanted to point out is doing a lot of activities. So you see here, Sarah was commenting, interacting with the community members we have here, not only just your group members, but with others in the community. And, then as she was talking to somebody, they replied to her, and, they said that, how they were loving the courses, and Sarah shared her view. So that is a very good way of interacting with the community, because you never know where opportunities arise, who you meet in this platform. We are such a mixed bunch of people. We are students, we are researchers, we are scientists, and all of us are learning together, working together, and doing our research together and learning more. So this is a really very good way of seeing what you can do with the platform. And the more you leverage the profile, the more benefit you get out of it. So moving on, the organizational page, as we have already always spoken about, is you can see that we are right now on 13th January, and the next is coming up on 16th. So right now I just posted here the free webinar that we had last day. So anybody who is in the organization page can see this. You can put a favorite here. So see, I have favorited a few people, so it's showing up like, you can also remove your favorite. And the more you do all of these activities, you get like, 10 points for doing, updating your profile, or doing a favorite on any of those activities that other users around you are doing. And it is also a great way for us to see the progress. So right now, as none of us have really started, you see the education and server use is like 0, but I'm hopeful that, by the next class, if some of you start taking the introduction to bioinformatics courses, we will see some progress on the education, and it will all be updated here. For others whose names are still not here, we'll be updating it by the next class, and, we can do the courses. And you also start to see that, the progress section, we see that there is a red bar, because from today the event is starting. So once you start taking all of these courses and, points start appearing here, we'll be seeing that progress bar moving forward. So there'll be a graph, and you can check on the progress of every member here. You can check on your individual progress. And I also wanted to point out like a few profiles. So you see here, Shritama, she has been like updating her profile. So that is very good. Shritama, she has her complete name. She has the Amity logo, and she also collected 200 points right now. So she's already starting. She's off to a good start. And that is like a very good thing. And then we have Priyanshu. Priyanshu, I hope that you will be doing it by the next class, update a profile, put in some links. Shritama, you could put in some links to your profile, and you'll be starting. So once you create an account, you just get 100 points instantly. So that is like a really nice way to start. The more points you collect, the more courses you do, the faster you rise up in the leaderboard, which we had talked about in the previous session we had. And so Shinjini, Shinjini's profile has to be completed now, and I would also suggest that, Shinjini, you can take that dot off. So this is just your name, right? This is how you would want people to address you, see you, and know you by. So instead of using any kind of usernames that are generally be used for all of this different social media activities, you can actually go ahead and update your profile. So that would be a very nice way. And this was Komolika. So Komolika has also like updated her profile, and she again has 200 points. Now we do not have so much of time to go ahead and talk about each of your profiles, and maybe we'll do so in the next classes, and we will see some progress. All of you who have not received your TBio login info should be receiving them very soon before the start of the next class. And I'll stop sharing my screen right now. And thank you so much, everybody. Mohit, you can just.
Thank you, Bipka. Thank you for that wonderful um, overview of how we should be, taking care of our profiles and how it's going to help us. So I'll just quickly go to the next slide, which is like the assignments. So we already have talked about it. To complete your profile with an image, explore the courses and project under the Bioinfo platform. So these are two simple assignments that you have to do, and it will be really nice to see the progress. Upcoming workshops, we would like to tell you that what is going on, what is happening, and what are the opportunities next. So we'll be having the next session on 16th. Looking forward for you, all of you, to be there and learn more about the transcriptomics data, where we will be going in a little more depth, and with your knowledge and with the educational access, I think we'll have more questions. We are going to start a new program, omics logic transcriptomics, tomorrow. So it's an online program. It will be open for those people who have registered for it, but it will be great to see. I mean, if you guys are interested in seeing the first class, we'll be happy to share the link. So I would like to thank everyone today who has come and joined this this meeting, and have something to add, and have something to learn, and have something to look forward for the next class. So I'll be open for question and answer right now. So if you have any questions, please ask us, and we'll be happy to answer those. Hi, Nitin. I think Nitin has opened his mic. So do you have something to ask?
No, right now.
Okay.
First of all, I will go through all the courses that you have told right now.
Sure, that would be great. I mean, if we can, quickly, do this and then move on, and then we'll learn more, and we'll keep on doing that throughout this course. So I'm really glad and pleased to have you all here, and we are really happy to start this experience and start this journey together. And we're looking forward for you to join us in the next class, and where we will learn more. And we'll also be emailing you, as I said, that we'll be emailing you the video recording of this session, and also the content, and whatever is educational material that you would require. Hello, Soham, do you have something to say?
Oh no, I don't have something to say. Just I was actually telling that Debushmita has raised a question in the chat box.
Okay, can you? Okay.
I have answered Deboshmita. So this is something that I want to tell everybody, that there are some paid courses that you would see, paid projects that you would see. So if you're interested in that, you're very much interested in doing it, just let us know. You have the email ID, you have the WhatsApp group, just let us know and we would make it available for you. So those are by request. But first, it would be great if you could go with the coursework that is already there, so that we can see your progress, and we know that, okay, you are doing well and you can take up that course. So that way, you just let us know. Okay, I'll put all of our email IDs again in the chat. Yes, and thank you, Deboshmita.
Okay, thank you everyone for joining us. We'll be seeing you real soon then. Have a good night.